Good morning, class. Today we are going to talk on heme synthesis, right? How heme is going to be synthesized and what are the steps involved in that, and what is its regulation, different types of regulation, and what is the significance of that, followed by even the disorders which are related to heme synthesis. Okay, so you all know this is the structure of the heme, which is protoporphyrin 9 ring structure with the with the globin chains and even the protoporphyrin 9 ring structure at the center, which is stacked inside the globin chains. So there will be 4 globin chains, 2 alpha and 2 beta. Center you can see there is a pocket kind of the thing. In that pocket kind there is a heme is going to be lodged in, and heme consists of heme with iron in +2 condition, that is in the reduced condition. So this is a little bit about the chemistry, right? And then the important function of the heme is to gaseous exchange in the respiratory system, that is in the lungs. That is the primary function of the heme. So let us see now how heme is going to be synthesized. So this is the same what I was telling you. So it is consisting of 4 pyrrole rings, nitrogens of 4 pyrrole rings facing towards the center, and all these 4 nitrogen rings, atoms are being coordinated with iron, which is there at the center in +2 state, that is in ferrous state. Okay, and iron has 6 bonds. 4 are stabilized by 4 nitrogen atoms of 4 pyrrole rings, and one is for the oxygen, fifth one. Sixth one is with the imidazole ring of histidine on the globin chain, and sixth is coordinated with the oxygen for the gaseous transport. Biosynthesis of heme is going to be synthesized in two areas. One is in bone marrow erythroid cells, especially in normoblasts. So about 85% of the synthesis is going to take place here, but not in the matured ones. So it is only in the in normoblasts, that is in the bone marrow erythroid cells. Liver constitutes about 10%. Okay, but majority is going to be taking place in red bone marrow only. Fine. And the pathway is partially mitochondrial and partially cytoplasmic. Totally, it consists of 7 steps in this process, and step 1, 5, 6, and 7 are going to take place in mitochondria, and step 2, 3, and 4 are going to take place in cytoplasm. So this division is very important, that is partially mitochondrial and partially cytoplasmic. As I said, it is partly mitochondrial and partly cytoplasmic. First step is going to take place in mitochondria, right? So here there is a differentiation of that, how it is going to take place. So let us go stepwise. So it consists of 7 steps, step 1 to step 7, right? Step 1 is the synthesis of delta aminolevulinic acid. Step 2 is formation of porphobilinogen. Step 3 is formation of uroporphyrinogen. Step 4, formation of coproporphyrinogen. Step 5 is formation of protoporphyrinogen. Step 6 is formation of protoporphyrin, and finally last step, that is 7th step, is formation of heme. So let us try to see one after the other. Step 1 is synthesis of delta aminolevulinic acid. So it is going to be synthesized from 2 substrates. The first substrate is one of the simplest amino acid, that is glycine. Glycine condensing with one of the intermediate in in Krebs cycle, that is succinyl-CoA. Succinyl-CoA, the origin of the succinyl-CoA is from TCA cycle. So we all know TCA cycle is taking place in mitochondria. That is the reason why the first step is of heme synthesis also happens in mitochondria, because of the availability of the substrates, right? So glycine condensing with succinyl-CoA in the presence of the enzyme ALA synthase, that is aminolevulinic acid synthase, for which the needed coenzyme is pyridoxal phosphate, otherwise in short known as PLP. And the product formed here is delta aminolevulinic acid with the liberation of carbon dioxide and with the liberation of coenzyme A. Right? So in nutshell, glycine condensing with succinyl-CoA in the presence of the enzyme ALA synthase and a coenzyme, that is pyridoxal phosphate, forms into the first product, delta amino levulinic acid. Second step is formation of porphobilinogen. This is going to take place in cytoplasm. So delta amino levulinic acid is being carried from or transported from mitochondria into cytoplasm. Here in the cytoplasm, two molecules of ALA are going to be condensed to form into porphobilinogen by the removal of water molecules, and which is going to be catalyzed by the enzyme ALA dehydratase. Right? So ALA dehydratase is the enzyme. Two molecules of delta aminoleuvulinate are going to condense, liberating two water molecules, forming into the first monopyrrole, that is called as porphobilinogen. And this enzyme ALA dehydratase is zinc dependent, and which is going to be inhibited by lead. So this is the specificity of this particular step, formation of first monopyrrole, that is porphobilinogen, which is going to take place in cytoplasm. In nutshell, now combining the two steps together, glycine condensing with succinyl-CoA in the presence of the enzyme ALA synthase with the coenzyme pyridoxal phosphate. First intermediate, unstable intermediate formed is the alpha amino beta ketoadipate, and by the action of the same enzyme, again ALA synthase with the liberation of carbohydrate, it is carbon dioxide, it is going to form into ALA. Now this ALA from mitochondria comes into cytoplasm. In the cytoplasm, two molecules of ALA are going to be condensed with each other, forming in the presence of the enzyme ALA dehydratase, forming into first monopyrrole, that is the porphobilinogen. Right? And then step 3, formation of uroporphyrinogen. Now here 4 molecules of PBG, that is porphobilinogen, are going to condense to form into uroporphyrinogen. The pyrrole rings are joined together by methylene bridges, which are delivered from alpha carbon atom of glycine. And this is balanced by two enzymes. One enzyme is known as PBG deaminase or UPG1 synthase. The second enzyme is UPG3 co-synthase. Now let us try to see how these two enzymes are going to act independently. By the action of UPG1 synthase or PBG deaminase, 4 molecules of ammonia are going to be liberated, and a linear tetrapyrrole called as hydroxymethylbilane, that is HMB, is going to be formed, and this HMB spontaneously cyclizes to UPG1, right? And this UPG1 is going to be converted into UPG3 by the action of another enzyme, that is UPG3 co-synthase. These Roman letters are very important. The first Roman letter containing molecules are useless, which does not have any biological function in the body. Only 3 isoforms are going to be important in the synthesis. So UPG1 formed in small quantities are rapidly oxidized to uroporphyrin 1, and then it is going to be excreted in the urine. Right? So here 4 molecules of porphobilinogen are condensing with each other end to end in the presence of the enzyme UPG1 synthase or PBG deaminase. Initially it is going to form into a linear structured tetrapyrrole called as hydroxymethylbilane, which is going to be spontaneously cyclized to form into UPG1. This UPG1 is acted upon by the enzyme UPG3 co-synthase, finally to form into UPG3. This is uroporphyrinogen. Okay, don't try to write as uroporphobilinogen. Bilinogen is something different from porphyrinogen. The same one to put together again, to narrate, glycine condensing with succinyl-CoA in the presence of ALA synthase enzyme, and PLP is the coenzyme, forming into delta aminoleuvulinate. This delta aminoleuvulinate coming out from mitochondria or transported from mitochondria into cytoplasm, where 2 molecules of ALA are going to be condensed with each other in the presence of the enzyme ALA dehydratase to form into porphobilinogen. 4 units of 4 molecules of porphobilinogen condense with each other in the presence of the enzyme uroporphyrinogen 1 synthase to form into HMB first, that is a linear structure of hydroxymethylbilane, right? And which spontaneously cyclizes to form into UPG1. UPG1 is acted upon by the enzyme UPG3 co-synthase to form into UPG3. Now step 4 is the synthesis of CPG, that is coproporphyrinogen 3. 4 molecules of carbon dioxide are going to be removed from UPG3 by the action of the enzyme UPG decarboxylase, and the product formed here is CPG3, otherwise known as coproporphyrinogen 3. So here the decarboxylation reaction takes place, and the acetate groups which are present onto the independent pyrrole rings are going to be decarboxylated to form into methyl groups. So uroporphyrinogen 3 removes its 4 carbon dioxide units in the presence of the enzyme UPG decarboxylase to form into coproporphyrinogen 3 or CPG3. Further metabolism takes place in mitochondria. So step 2, 3 and 4, that is formation of PPG and UPG3 and CPG3 forms in cytoplasm. Now this particular CPG3 is now entering into mitochondria. In the mitochondria, CPG3 is oxidized to PPG3. PPG is 3, the full form of it is protoporphyrinogen 3 by the action of the enzyme CPG oxidase. So here 2 of the propionic side chains are oxidatively decarboxylated to form into vinyl groups. Okay, CPG oxidase act only on type 3 forms of CPG only. Okay, CPG3 acted upon by the enzymes CPG oxidase forms into protoporphyrin 9 or protoporphyrinogen 3. Right. Now generation of PP3. PPG3 is oxidized by the enzyme PPG oxidase to protoporphyrin 3 again in mitochondria. This oxidation requires molecular oxygen, and this reaction occurs in the liver. The methylene bridges are oxidized to methynyl bridges. Thus we are going to get the colored products. Thus formed final compound is protoporphyrin 9. And this protoporphyrin 9 gets the incorporation of the iron in the form of +2 iron by the action of the enzyme either by ferrochelatase, or the other name of the enzyme is heme synthase, which is located in mitochondria. Iron is being incorporated into the center of this molecule, finally forming into heme. Okay, now this iron is going to be coordinated, linked with the 5 nitrogen and 4 nitrogens of 4 pyrroles, and 5th nitrogen, 2 nitrogen atom of histidine residue, and 6th is opened for oxygen. So finally the step 7 gives rise to the generation of heme. So protoporphyrin 9 acted upon by the enzyme PPG oxidase gives rise to protoporphyrin 9, which is acted upon by the enzyme ferrochelatase, introduction of iron into the ring structure forms into heme. So in a nutshell, all the reactions put together. To summarize, glycine condensing with succinyl-CoA in the presence of the enzyme ALA synthase, liberating carbon dioxide and coenzyme A forms into the first product, that is ALA, delta amino levulinic acid. This step is taking place in mitochondria. From mitochondria, ALA comes into the cytoplasm, wherein in the cytoplasm 2 molecules of ALA are going to condense in the presence of ALA dehydratase enzyme to give rise to first monopyrrole, that is porphobilinogen. 4 units of porphobilinogen acted upon by 2 different enzymes. First one is UPG1 synthase forms into linear structure which cyclizes to form into ring structure. UPG1. UPG1 is acted upon by UPG3 co-synthase to form into UPG3. Now, UPG3 is acted upon by UPG decarboxylase to form into CPG3. CPG3 is acted upon by the enzyme CPG oxidase to form into PPG3. PPG3 acted upon by the enzyme PPG oxidase forming into protoporphyrin 9, which is acted upon by ferrochelatase to introduce iron in the +2 form into the ring to form into heme. So this is in a nutshell regarding the synthesis of heme. The same pathway, pictorial representation to represent first step is taking place in mitochondria. Then second, third, and fourth steps are taking place in mitochondria. Again, 5, 6, 7 are taking place in mitochondria. So total of 1, 5, 6, 7 are taking place in mitochondria. 2, 3, and 4 are taking place in cytoplasm. So which is the very common examination point of view question is this particular heme synthesis.